So the important thing about the approach that we use is if you take a bunch of different patients with cancer, of course, if you're going to be able to use this assay in a clinically useful way, you need to be able to find an answer in most patients. So it doesn't do you any good to have a test that only works for people 1 out of 10 times. And so an important component here has been to demonstrate that we can actually find the cells as they're spreading in most, if not all, but most patients with solid tumor cancers. So that's really what the first basic paper demonstrates. We took patients with all different kinds of cancer, and we showed that not only can we find these cells in most of those patients, far more than has been seen before, but also, and importantly, this is my angle on it, we can see them really, really well. These are not blurry little dots that we're counting. These are beautiful, crisp, high-resolution images that even a pathologist who spends her life looking through the microscope is happy with. I can see the nucleus, I can see the cytoplasm, I can see all the structures of the cell that I need to see to really feel like I'm able to investigate these cells with confidence.
And then one aspect that became immediately clear looking at the images was actually an observation that hadn't been made before around the fact that we find clusters of these cancer cells together with white blood cells in many of these patients. And that was something that we in that circulating tumor cell field hadn't really seen before. It was something that came from the fact of how we actually how we run the technology approach, and it was for us a great finding technically, but then of course we threw that over into Kelly's world, and we're like,
right, and this is really interesting to us over on the diagnostic side, because we know that when someone has a cancer in a solid organ and then they get metastatic lesions, they don't get usually hundreds and hundreds of metastatic lesions. So not every cell that leaves that primary tumor and goes out into the bloodstream, it must not everyone ends up forming a metastatic tumor. So what makes a person have 3 sites of metastasis? They have a primary tumor removed, and then they come up with 1 nodule in their liver and 1 nodule in their brain and 1 nodule in their lung. Why only 3? Because we know that thousands of cells must have left that primary tumor. So this idea that there's a difference between the types of cells that are in the bloodstream, and we don't know exactly what the important differences are yet. That's what we're working on. And it could be that these clusters, maybe they're quite different than individual cells. They have friends, they have other things with them that might make them more likely to survive in a foreign soil where they would land after their trip through the bloodstream. So we're very excited about that. And other differences that we're finding between these cells. They're not all the same. Some are probably dying, some are much more alive, some come in clusters. It's going to be very interesting to see which ones turn out to be the ones that we care about.
So that was the second paper. The third paper then really came about through our collaboration with 2 lung oncologists. And finding these cells in lung cancers has been a real challenge for the field. And so we set out to address that after we had seen the success we had in the other cancer types. And we figured out a way of how to do this in lung cancer patients as well, and then followed that immediately with 2 particular studies where the first one just tries to figure out, do we find them in a majority of the patients, as Kelly described before, and the answer to that is absolutely yes. And then that is paper number 3. Paper number 4 then asked the question, if we take blood samples through the course of the disease, say over a 6-month period, what changes? Question 1, can we find them again and again? And then question 2, what changes? And the answer to both of these questions is, of course, yes, we can find them again and again. And the number of these cells goes up over time, which again makes sense from a disease progression perspective. And that is, of course, the beginning of this overall program. The last paper then describes the first time that we, in this particular approach, then applied what we call a biomarker on top of this fluid biopsy. So in this case, we worked together with an oncologist at the University of Southern California up in Los Angeles, and developed an assay that looks at androgen receptor, which is a key driver in prostate cancer, and tried to figure out, can we actually find that androgen receptor on the cells that we identify in all of the patients with prostate cancer, and that is what's described in that last final paper. We find androgen receptor being expressed and localized to the nucleus. This comes back to Kelly's earlier comment about the quality of the imagery, that you can clearly identify where in the cell that particular protein is expressed. So we found these cells, they express androgen receptor in the nucleus, and in any given patient, of course, you have a big heterogeneity between cells that express the androgen receptor, cells that don't express the androgen receptors. Then you have patients that have lots of the positive cells, and you have patients that have none of these positive cells. So it is that is the beginning of a much larger study in prostate cancer, where we're following up on that work now.